Hi guys, this is Mrs. Ulmer, and today we'll be talking about non-Mendelian types of dominance. So before we get into the non-Mendelian types, let's remind ourselves of what Mendel said. So when Mendel studied his pea plants, he came up with his idea of complete dominance, in which one allele is completely dominant over the other allele. So when he crossed the red with the white flower, he gets red because red is completely dominant over the other. So the two phenotypes were red, the dominant one, and white, the recessive. So this is what we've been doing for the last few days. As a reminder, the heterozygous genotype, in this case big R, little r, will always have the dominant phenotype. So heterozygous is dominant. And there are only two phenotypes. That's the big clue here when it follows Mendel's law, is that you'll only see two phenotypes, the dominant phenotype and the recessive phenotype. So again, complete dominance, what Mendel told us, one allele is dominant over the other. There's only two phenotypes, the dominant and the recessive, and the heterozygous will have the dominant phenotype. Now, the first type of dominance that doesn't follow the laws of Mendel is called codominance. In codominance, both alleles are dominant. So in this cross, we have a red flower and a white flower. But what's different here is that red and white are both dominant. So instead of seeing red in our offspring, we see red and white. Both are dominant, so both show up in the outcome. And so when we have a heterozygous genotype, so in this case, again, it would be RW, one of each allele, that heterozygous now shows both phenotypes. And notice it shows them separately. You'll see part that are red and part that is white. So it shows them separately. So codominant, both are dominant. So you'll notice we treat the letters a little differently as well. So the capital R represents the red allele, and to be red, you have to be big R, big R. The capital W represents the white allele, and to be white, you have to be WW. And if you have one of each, you have that heterozygous genotypes, you're red and white. That and is the big giveaway because both of those are showing up separately. So for this type of dominance, codominance, we have an example. Here is a black chicken, which would be big B, big B. So again, B is the black allele, but to have that black color in your feathers, you have to have two big Bs. The white is WW. So the white allele is W, and to be white, they have to have big W, big W. But if you get one of each, because it's codominant and both show up, we see this speckled or striped pattern in the chicken feathers. So we're going to cross a black chicken, big B, with a speckled chicken, big B, big W. And we're going to analyze the results of this cross. So take a minute to answer the question of how many are black, white, and speckled. So if you answered these correctly, you got 2 out of 4, or 50%, that are black, so they have big B, big B. 0 out of 4 are white because none of them are WW. And 2 out of 4 are speckled, big B, big W. So again, in codominance, both alleles are dominant. So both are going to show up separately. So you're going to see that and, this and that show up in the heterozygous. So both show up when you have one of each letter. All right, the second type of dominance that doesn't follow the laws of Mendel is called incomplete dominant. Now, whereas codominant, they're both dominant, and incomplete dominance, neither allele is dominant. They actually act recessive. And so when you get to the heterozygous, neither of them dominate, and instead you see a blending of both of the alleles. So in this case, we have our red and white flower again, but in this case, they blend together to make pink. So for incomplete dominance, I always think about throwing them in the blender, and the heterozygous is a blend. Now, again, we use those capital letters to represent the allele, and we use separate letters. One R for red. So to be a red flower, you have to have big R, big R. To be white, you have WW. And then when we blend the two, big R, big W, or big W, big R, you get pink. We threw it in the blender. That's incomplete dominance. So if you check this out, in flowers, we have big B, which is blue, big R, which is red. But notice when we're heterozygous, we have one of each letter, big R and a big B, it's purple. That's the blending of red and blue. So in the Punnett square here, I'm crossing a blue plant with a red plant. So take a minute to answer the questions about this Punnett square between these two flowers. Okay, if you answered correctly, you got 0% were blue and 100% were purple, because all the offspring were capital B, capital R, which is that purple genotype. So in incomplete dominance, neither allele is dominant, and neither really show up in that final heterozygous product. So for heterozygous genotypes, one of each letter, we throw it in the blender. We blend the two together to get our heterozygous phenotype. So let's do some practice with this. So just as a reminder, for Mendel, he had complete dominance. So if we had curly and straight, he said curly was dominant over straight. So if you got one of each, you would be curly. For codominance, we have curly and straight, but the final outcome, if it's codominant and they're both dominant, we would see some parts curly and some parts straight. So, and both of them would have to be there. For incomplete dominance, neither is dominant. So we're going to see curly and straight. When we get that heterozygous, we'll see kind of a wave, a blend between the two. So for incomplete, we throw it in the blender. For codominance, both show up. They're both dominant. They both show up. So let's do some practice. So for each scenario, you're going to determine, is it complete dominance, what Mendel said? Is it codominant, both are dominant? Or is it incomplete dominance where we threw them in the blender and they were a blend? So answer this question. Fish can be red, blue, or red and blue. What type of dominance is this? All right, if you answered that this was codominance because both showed up, the red and the blue, you were correct. Try this question. Flamingos can be pink or purple. So when I cross the pink with the purple flamingo, the babies are pink. There are only two phenotypes here. So is this codominance, incomplete dominance, what Mendel said and told us about complete dominance? All right, this is an example of complete dominance. Notice that one was completely dominant over the other. So there are only two phenotypes, pink and purple. One wins when they're blended together in that heterozygous. All right, next question. Horses can be black, white, or gray. So what type of dominance is this? Notice that neither trait showed up in the heterozygous. It's a blend here. Codominance, incomplete dominance, or complete dominance. If you had answered incomplete dominance, you were correct. We threw it in the blender. Neither allele was dominant. Neither fully shows up. Throw it in the blender to get that gray, a mix between white and black. All right, here's the next example. Birds can be blue, tan, or blue and tan. So we see the blue and we see the tan separately. So what type of dominance is this? If you answered codominance, both are dominant in this case. Both show up in that heterozygous. Both showed up here. That's codominance. Both are dominant. All right, that was the last one. So if you have any questions, just let me know and have a good rest of your day.